Most of you are probably watching this video from home, worried about the novel coronavirus, which, on the surface level, seems to have outsmarted humanity. There are no confirmed drugs and no tested virus in the market for this disease. And every day, as I pull up the coronavirus test cases in Massachusetts, I almost know that they would have increased by 100, doubled, or maybe even tripled. But here's the question, are we really that powerless? Will we have to wait till 40 to 60% of the United States is infected till we can stop this pandemic? Over the past few days, I've been doing deep dives into the different efforts to combat this pandemic, from NextStrain's efforts to map the evolution and the pathogenetic mutations of the virus, to a new breakthrough study which allows us to use machine learning to program Cas9's cousin, Cas13, to make knockdowns in the viral RNA of SARS-CoV-2. And the efforts extend much closer to home as well, from apps that can help prevent us from crossing the paths of other people with the virus, to machine learning algorithms that can distinguish between symptoms of the coronavirus and the flu. Scientists and researchers are beginning to find ways to get us through this hard time by leveraging exponential technologies. And the technology at the center of all of this, artificial intelligence. It's allowing us to take geographical data, telehealth data, and make meaningful predictions that could ultimately save lives. But that's not it. It's also allowing us to generate potential drugs that just could put most of this to an end. Recently, I was able to repurpose a generative drug design algorithm made by Topaz Ape on GitHub, which served as an implementation of a paper published years ago by Gupta et al. Ultimately, it takes the structure of a drug molecule, generates variations of them, and then it allowed me to test the potency by using other software. But before we get to all of this, we have to understand how a drug works. Drugs are small molecules that either activate or inhibit three-dimensional protein or nucleic acid structures. You can kind of think of them like a lock and key. The small molecule is a chemical drug that needs to have just the right properties and shapes so that it can fit into the grooves of the protein. And in order to create a small molecule drug for SARS-CoV-2, our first goal would be to determine a protein target. Determining a protein target involves looking at the viral life cycle and finding the best ways to sabotage its ability to replicate and infect nearby cells and people around it. And the way to do this is by finding the set of macromolecules which without the virus cannot function. So let's look at how the virus functions. The coronavirus is what's known as a positive-stranded retrovirus, and it inserts its genome into the cell. The virus's genome consists of two open reading frames, which are essentially two ways that the virus can translate its genome into proteins. The genome is first translated into a polyprotein, which consists of all of the functional proteins of the virus. And it's kind of like beads on a string. Next, we need to look at which of these beads we can remove. So which of these functional proteins we can remove so that the virus cannot replicate and cannot infect other nearby cells around it. So after reading through a few research papers, I was able to identify 3C-like protease as an effective target, which I could inhibit with a small molecule drug generated by AI. 3C-like protease's job is to act like a pair of scissors, which essentially break down the long necklace of proteins into each of the individual proteins that allow the virus to replicate. So therefore, by preventing 3C protease from acting on the polyprotein, we prevent the virus from replicating. Now on to training our machine learning model. The model which I used is called an LSTM, or Long Short-Term Memory Network. Think of the machine learning LSTM as a preschooler learning to be proficient in English. First, it reads a particular sequence or text to understand patterns or structure in the text. Similarly, each unit in LSTM does this by taking input from a particular element in a sequence and then passing on this information to the next cell in the sequence. The next cell takes its input along with the hidden state to make the prediction about the next element in the sequence through something called the softmax function. The LSTM can do this exact same process on what we call smile strings, which are one-dimensional representations of two-dimensional chemical molecules. The way that I trained the neural network to read is by downloading millions of chemical sequences from the Chembl database, and I trained my LSTM by asking it to predict the last n characters given the first n, in essence to predict the next character. I used SQL to select small molecules from the database with particular metrics, and then tokenized and one-hot encoded the sequence to represent each character as zeros or ones. So the first thing I did, after downloading my millions of molecules from the Campbell database, was I had to tokenize these molecules and one-hot encode them. And what that means is that for every unique character in this smell string, I had to assign it a value, so 1, 2, 3, 4, etc. And then using those assigned values, I had to want to encode each character, meaning each character is represented by an array of zeros and ones, where the position of the one represents the character, which is being one-hot encoded. So after one-hot encoding, where the code you can see right here, so the tokenized function, I was then able to actually go and train my model using the hyperparameters in my configuration file. So these are the different hyperparameters that I use to train my model in my configuration file. And so I trained over 22 epochs. My LSTM had 256 cells or units, and I used the common atom optimizer with a batch size of 256. My validation split was 0.1, so for every 1 out of 10 smell strings, that one smell string was used for validation. And here you can see something called the sampling temperature. So essentially what the sampling temperature is a factor that is divided by for every element of the softmax function, and increasing the sampling temperature increases the diversity of the molecules that can be produced. Here is the actual model itself, which consists of 2 LSTM layers with a dense layer at the end and a softmax activation, which is actually going to predict the next character in the sequence. So now that we have trained our model, we can actually generate variations and generate samples. So here you can see that I generated 30,000 variations of those molecules which I trained the LSTM on. And what happens after it generated those molecules is it has to make sure that the molecules that it generated are actually valid molecules that would exist or could exist in nature or man-made. So here we can see that it generated 63 valid molecules. But the good thing is for each of those valid molecules, if we backtrack and we find the smile string for each of those molecules, the smile strings all made sense. So now we're set to go because we've filtered the molecules that would actually exist or could actually be prepared by medicinal chemists. So here essentially what I'm doing is I'm creating 2 sets of chemical fingerprints. And what fingerprints are matrices of zeros and ones that represent the different atomic interactions and different elements of structure within a molecule. So they're different from the smile strings. So what we do is we create a set of fingerprints for both the generated molecules and the original ones. So here, OFPS, so this set of fingerprints is the set of fingerprints for the original molecules, whereas this set of fingerprints is the set of fingerprints for the generated ones. And here you can see what a fingerprint looks like. So it's an array of zeros and ones. So then what we can do is using our chemical fingerprints, which essentially keep tabs on the properties of the molecules and the different interactions and elements of structure that are taking place, we can do a principal component analysis. And principal component analysis is taking all of these different dimensions to the fingerprints, all of the different interactions, and so on, and it's condensing that down into 2 dimensions so we can easily interpret whether overall the properties of the generated and original molecules are similar in nature. And as we can see here, there is quite an overlap between the generated ones, which are in blue, and the original ones, which are white circles. So we're good to go because the model was able to make different molecules while at the same time keeping the properties and nature of the molecules intact. And another way we can measure more specifically for specific properties, like molecular weight and log p, which is essentially a measure of how much affinity the molecules have for water or nonpolar substances, and where they fall on that scale. We can look at violin plots. So a violin plot is essentially a distribution for the molecules where the largest horizontal width corresponds with a higher frequency. So here we can see here the violin plots for the generated and original molecules are again very similar. Not much of a deviation, a significant deviation that makes us reconsider the model's accuracy. So here we can see what one of the smile strings looks like. So of the generated molecule, and a smile string is again is just a sequence of different characters that are literally representing the structure of the molecule. So unlike fingerprints, bit fingerprints. So here we can see the different elements: oxygen, carbon, bromine, and they're all being represented in this chemical ring structure. So this is the one-hot encoding for each of the elements in the smile string. So going back to what we talked about initially, you can see each of the characters and their representation with an array of zeros and ones. And yeah, that's essentially how we were able to teach the model how to read the language of chemical molecules and just understand general patterns and general structure in our data for smile strings. Next, what we're going to do is we're going to take this to the next level by having it generate variations of molecules that specifically bind to the 3CL protease receptor. So here we are making variations of the representative molecules which had a high binding affinity for the SARS-CoV-3CL protease receptor. And the way that we train our model this time and have it generate variations is very similar to the first time, except this time we're just loading a different set of data, but all the tokenizing processes and one-hot encoding stays exactly the same. So here we can see that the model is training for 10 epochs, and each time the loss is going down, which is great. So that means the model is able to accurately predict the next character in each of the fine-tuning smile strings. And once we have effectively trained our model, we can actually have it generate around 100 different variations and different molecules. And now what we have to do is we have to compare the original set of representative molecules that we trained, the generated molecules, and all of the SARS-CoV inhibitors, all on one plot, just to see how all of their properties compare. And so just like last time, we make a separate set of chemical fingerprints for each set of molecules. So for the known inhibitors, so all of the known inhibitors, not just the ones with a high affinity, we make one set of fingerprints. And then for the set of inhibitors that were representative and that we actually trained on for the fine-tuning, we create another set of fingerprints. And finally, we create a last set of fingerprints for all of the SARS inhibitors. So now what we can do is we can, again, do PCA analysis and plot all of these and see how they compare. And overall, we can see that the selected training molecules, so the representative ones represented by stars, the fine-tuned ones, so that means the ones that were generated, and then the white circles, so the ones which are in the background, all have significant overlap in terms of properties. And the way we can more quantitatively assess this overlap is through something called the Tanimoto similarity. And the Tanimoto similarity essentially is using the bit vectors of zeros and ones and comparing their intersections to assess how similar the molecules are. And what we can do is we can print the Tanimoto similarity, and what we see is the molecules generated have a very high similarity to the original one, so around 0.93 on a scale of 0 to 1. And the highest, as we can see here, is actually 0.94, so really high. And what we can do after that, what we can do is we can compare the generated molecules to the original ones side by side, and we can see that the machine learning model changed simple things like a halogen from a chlorine to a bromine, or just adding what looks like a phenyl group with a chlorine substituent. But as we go down, it starts to get a little more complex, but overall the similarities are very, very high, and the structures are relatively close to each other. So then what I did is I converted each of these molecule structures back to the SMILE structures, and now what I had to do is I had to take these SMILE strings and convert them into what's known as SDF files, which would be used for the small molecule docking, which I would do next to actually test whether binding affinities for the molecules were stronger than the original ones. I used a software called PyRX or Autodoc Fina to test the binding affinity of the molecules. So first I had to convert their file format from a molecular programming object into SDF file. I also downloaded the crystal structure of the 2019 novel coronavirus 3C-like proteinase, as researchers in China had just discovered it a few days ago. First, I lowered the energy conformations of the small molecules and then selected a small window which restricted the binding of the small molecules to the active site of the protein. And I found the active site of the protein by doing research on the active amino acids that were involved in the enzymatic function of 3C-like protease. So now is the big moment. I tested each pair of molecules and then compared their binding affinities. In some cases, the machine learning model actually made some molecules with higher binding affinities than the original ones. So a few days after I finished my project, I was surprised to see that other drug discovery companies had taken similar approaches. InSilico had just released a white paper detailing how InSilico had used 28 machine learning models to determine the efficacy of new generated drugs, and had appealed to medicinal chemists to validate these. Turns out they had used the exact same target that I had, 3C-like protease. So let's move past machine learning. IBM's supercomputer has just been able to detect more than 77 potential molecules. That's insane. We're literally leveraging exponential technologies to do more than we ever could and ever have done in the past. Normally, drug discovery takes at least 1 to 2 decades, but using these technologies, we can accelerate past the first 5 to 10 years and get treatments to patients fast. At the end of the day, it isn't about hype. It's about how we can make the situation better using the technologies that we have access to. Quantum computing, artificial intelligence, they aren't just buzzwords. They're technologies that we can leverage to make drugs to get rid of this pandemic and save lives of hundreds of thousands of people. And to be frank, the COVID situation seems to be getting worse day by day. I know that COVID numbers are only expected to go up from now. Ultimately, though, I think it's comforting to know that humanity has the tools it needs to fight and solve these problems.